Chapter 2, the chemistry of life. So what are the main student learning outcomes for this chapter? We have to remind ourselves about what matter is. See, all life is made of matter. It can either be living matter or dead matter, but it's still matter. So we need to study the atomic structure. So that would be the interrelationship between protons and neutrons and electrons, which are the subatomic particles. We should also review when an atom is stable. If it's not stable, then it will start to be very reactive. It will either donate or accept an electron from a different element. We will talk about the periodic table, which is a list of all the elements in the world. And it's these elements that will come together to form molecules. How do they come together? It's by chemical bonding. So we'll study the most important bonds that we see in biology. And after we study that, we'll see how it makes up the macromolecules in biology. So there are 4 main macromolecules. You can think of macromolecules, because it's present in food. It's a very good to have this in mind. Like if you take food, right, it's made up of either carbohydrate, fat or lipid, or protein. It also has some nucleic acids in the form of DNA and RNA, which we usually don't talk about. So you can have this as a guide as we go through the chapter. So we'll start with the structure of an atom. So all life is made up of matter. And if you zoom into the matter, it is made up of atoms. So here's the structure of an atom. And this is called Bose model of the atom. So if you take an atom, it is the smallest unit of matter that has the characteristic properties of that special element, chemical element. So when you say matter, it will have weight, and then it will also occupy space. And it can either be living or non-living. And inside the atom, so atoms will make up all the matter in the world. So in the center, we call it the nucleus. We have the positively charged protons and the neutrally charged neutrons. And zooming around it in fixed shells, like shell 1, shell 2, and so forth, we have the electrons. Electrons are negatively charged. So usually the number of protons will be very same to the number of the electrons. Then the atom is stable. If not, it will be called as an isotope. That we'll see it in the next slide. So when you're representing an atom, the chemical symbol for oxygen, it is O. For carbon, it will be C. The number of protons it's called as the atomic number. So it's written on top. And here's the element name. It's called oxygen. And the atomic mass is the weight of both the protons and the neutrons in the nucleus. That is the atomic mass. So each element will have both these numbers. Write it down as the lecture proceeds, because writing is a very powerful tool. It will help you to remember when you see words in your own handwriting. That's why I'm going very slowly, so that you can take the key points for every slide. The PowerPoint is also available on Moodle. So in the Bose model, where you can see the electrons zooming around it, it's not spherical as this picture shows. Now there's so many discoveries that's been made in quantum physics that show that these orbits can either be dumbbell shaped and so forth. So usually the number of protons and neutrons will be the same. Let's say they differ. It is then it will be called as an isotope. For example, carbon comes in a few isotopes. There's carbon 12 and 13 and 14. 14 is actually very radioactive because of this, they will emit radiation, and it has a lot of use in biology. It is used in carbon dating. When you want to find the age of a fossil, they will study the decay rate of carbon. And see here, there's a picture in your book that shows about a pygmy mammoth. And they're saying it's about less than 50,000 years ago, it roamed the earth. It also has a lot of uses in medical diagnosis. For disease diagnosis, they use isotopes. So isotopes will have different neutron numbers. For carbon-12, it has the same number of protons and neutrons, but in carbon-13, there's one extra neutron. Carbon-14 will have 2 extra neutrons. I forgot to mention something here in the Bose model of atoms. The first shell can hold 2 electrons. So here's helium. It's holding 2 electrons. The first shell is complete. So it is very stable. If it is not complete, then it will become very reactive. The second shell can hold up to 8 electrons. So different shells will have different energy levels, and they'll hold different number of electrons. The shell that is further away from the nucleus will have the most energy. And the shell orbit or shell closer to the nucleus will have the least energy. So they don't have to spin as fast as the ones that are further away. And here is the periodic table. So it's a display of all the chemical elements that has been discovered. 92 of them are naturally occurring. There's a total of 118. So 92 is occurring naturally. The rest are man-made in a lab. And here they are, there's like groups and columns here, which is called periods. And if you study the key in the bottom, it shows the pale blue are nonmetals. The green would be alkalis. And then there's transition metals, halogens, noble gases, actinides, and so forth. So they are kind of, these are not reactive at all. That's why they're called noble gases. So depending on the position in the periodic table, you can guess the property of that element. And of course, the atomic number and the atomic mass and the symbol for every element is shown. So this is a very interesting picture showing about what are the elements that you find in the human body. And it's very interesting to note that C, H, O, and N, oxygen, carbon, hydrogen, and nitrogen make the most of the human body. Your body is 61% oxygen, 23% carbon, 10% hydrogen and nitrogen. And then of course, there's also calcium, phosphorus. They are all the macro elements in your body. So even though there's 118 elements in the periodic table, it looks like our body is mainly made of these elements here. So it's very clear that our diet should be rich in all of this. So these are the macronutrients. And then the ones that you need in smaller quantities, it will be called micronutrients, like iron and all that. And then there's some that has to be found in very trace amounts in our body, because they are like help enzymes work. Like for example, iodine, you need very little of it. So they are called trace elements. So for an optical health, you need all these elements, and it can only come from food. So I already mentioned this slide here. So this is how it is represented. You know, the bigger number is the mass number. This is the atomic number. And here is the symbol, and here's the word to represent. So this picture is showing the different shells, the first shell, second shell, third shell, and so forth. This is very interesting. Remember, I told you the electrons in the innermost shell have less energy, whereas the electrons that are zooming in the outermost shell have a lot of energy. So if they should jump from the outer shell to the inner shell, it will give off a photon of light, which is very interesting to understand. So if it's going from a high energy to low energy, it will give off a flash of light energy. If it is going from the inside to the outside, you have to apply energy to the system. So Niels Bohr's model of the atom in 1913 is a very important discovery. The electrons are kept in orbit because of the positive charge of the protons. The protons are positively charged, so they exert an attraction pull, just like how Earth is keeping the moon in orbit because of the gravity of the Earth, right? The moon doesn't go flying away into space because the Earth is pulling the moon. So that's a good analogy to think of when you think of Niels Bohr's atomic model. All matter is made of atoms. It's called as the atomic theory. So here, the first shell can hold a maximum of 2 electrons. If there is only 1 electron, then that atom is very reactive because it's constantly looking for another electron. The second shell can hold up to 8 electrons. So that's why it's called octet rule. Anything less than 8, the atom is not stable. So it is going to be very, very reactive. So it is 2, 8, 8. Third shell is also 8. And I told you that the electrons are not running around the nucleus in perfect circles, but they can take all these different orbital shapes. If the innermost shell has 2 orbitals, right? Like innermost shell can hold 2 electrons. That means in a shell, there are 2 orbits. Second one will have 8 orbits in the second shell. So in the second shell, if atoms have fewer than 8, then it's going to be very, very reactive. So in general, so it's written out here, if 3 or less, it will have a tendency to donate the electrons. It's easy to donate than to go look for 5 more electrons. The number of electrons in the outermost shell is called as valence electrons. So now when an atom is not stable, then this is where it's going to make all the bonding. In order to become stable, it is either going to lose an electron or gain an electron. So bonds are the attractions that is caused by electrons in the outermost shell, which is the valence electron. So the process of making a bond is what is called as a chemical reaction. If atoms, what is a molecule? So molecule is 2 or more atoms are coming together. So if it is the same one, it is still called as an element. You know, there are 2 atoms of oxygen, it's called oxygen element, hydrogen element, nitrogen element. But if it's 2 different elements coming together to form a stable entity, it is called as a compound. So carbon dioxide has 1 carbon and 2 oxygen. Water has 2 hydrogen and 1 oxygen. Glucose, C6H12O6, has 6 carbon, 12 hydrogen, and 6 oxygen. So the most common bonds, so all atoms are trying to get into a stable state. So that is why they're forming all these bonds that we call as reactions. So the 3 common bond in biology is covalent bond, ionic bond, and hydrogen bond. So we'll see each one of them. What is a covalent bond? Co means, see that word co comes from some kind of a corporation, you know, a cooperative corporation. So they're sharing the valence electrons. That's why it's covalent. So covalent bonds are happening between electrons in the outermost shell. Usually they are found horizontally together in the periodic table, these ones that form the covalent bond. So here is an oxygen molecule. In the second shell, it has 6 carbon atoms. And in order to become stable, it is going to look for 2 more electrons. So it will share 2 electrons from the hydrogen. Hydrogen is also trying to get into a stable state. So it will share an electron with the oxygen, and this hydrogen is also sharing an electron with the oxygen, forming a covalent bond. So in water, the bond between oxygen and hydrogen is called covalent bond. So, however, oxygen is very, very electronegative. That means the pull for electrons is higher. So what happens is oxygen will pull the hydrogen towards it at a very great force. So because of that, there is a negative charge where the oxygen is and a positive charge where the hydrogen is. So this is called polar covalent bond because of the unequal sharing of electrons, because oxygen is electronegative. If it was equal sharing, it will be called as non-polar. Oxygen also, 2 oxygen molecules are coming together and they are sharing 2 electrons. So it forms a double bond here. So if you share one, it is a single bond. So 1 hydrogen and 1 hydrogen. If you're sharing 2 electrons, it's denoted as a double bond. So remember that most of the bond, it's a very strong bond. Covalent bond is a strong bond. Ionic bond is the bond between ions. Ions are atoms that have a charge. So this ionic bond will happen between elements on the opposite side of the periodic table. If you take sodium in the second shell, it has just 1 electron. So in order for this to become stable, it needs 7 more, which is very energy consuming. So what sodium will do is, it is better for it to donate an electron in order to become stable. Chlorine atom, on the other hand, has 7 electrons in the second shell and it needs just 1 more. So instead of losing 7 electrons to become stable, it is going to accept an electron. So sodium is going to lose an electron and become positively charged, Na+. Chlorine is going to accept an electron and become negatively charged. So this attraction between ions, positive charge is cation, negative charge is anion. So this charge between ions is called as the ionic bond. This is also a strong bond. A very common example is the salt that we use in our food, sodium chloride. The molecules are held together by ionic bonds. The next is hydrogen bond. So a very good way to understand hydrogen bonds is water. It's again interesting to note that 71% of our earth is water and 70% of your body is also water. And water is so important because life is sustained by water. It creates an environment that is sustaining life, nurturing life for all living things on earth. No water, no life. See, that's why when NASA sends out, like Curiosity rover to Mars, the first thing the machine is looking for is water, because for evolution to happen, we need, it happens in a medium of water. See, you and I need a lot of water to stay alive because all chemical reactions happen in a medium of water. So if you take water, remember I told you is it has a positive charge where the hydrogen is and negative charge where the oxygen is. And it's like a little magnet. It's going to attract 4 other molecules of water. And the bond between the positive hydrogen and the negative water is called as the hydrogen bond. So this is the third bond. It is a very weak bond. It's not very strong, but it forms very easily. It will break and form easily. And it's because of this hydrogen bonds that we see the unique properties of water. So let's briefly look at the properties of water. So water is an element that can, a compound that can exist in 3 different forms, right? Solid, liquid, and vapor. So when it freezes over, the hydrogen bonds will form a lattice structure like this, hexagonal crystal lattice structure. And because all the atoms are further away, ice is less dense than water. So in winter, during winter, you can see ice floating on top of the lake, thereby freezing the lake and insulating the lake. So what happens is even it becomes very, very cold outside, like -30 and so forth. Inside, under the ice, it's still warm and the marine ecosystem will survive. And it's this hydrogen bond in water is vital for life. If water was not freezing, right, like this, there won't be life on Earth, because everything, the whole ecosystem will collapse. Because once all the marine ecosystem dies, it's like a domino effect. It is a universal solvent. You know that, morning coffee, you want to put sugar, boom, sugar will dissolve in it. Put salt in soup, the salt will dissolve in it. It is a universal solvent. So things that can easily dissolve is in water, they're called hydrophilic. However, if you add like butter or oil to water, it doesn't dissolve because it's hydrophobic. So here they've added salt, sodium chloride into water, and water is non-polar. So what it's going to do is the chloride is it will break down into its ions. And because chlorine is negative, all the positively charged hydrogen will form a sphere around it. And the opposite for the sodium element. Oxygen will gather around it because oxygen is very, very negative. And this is the science behind how things dissolve very quickly in water. That's why water is so widely used in cooking, because everything can dissolve very quickly in water. It is a universal solvent. It has very high heat capacity. What does that mean? That means you have to add a lot of heat into the system in order for it to increase its temperature by 1 degree. And because of its high heat capacity, it's a great moderator of temperature. See, because you and I are made up of 60 to 70% of our body is water. Your brain actually is more water. Your blood is like a lot of water in your blood also. All the chemical reactions, right, happen in an aqueous environment. But because of the high heat capacity of water, it can help to maintain homeostasis in our body, which is 37 degrees centigrade. So when you become very hot, let's say you go on a hike or you exercise and your body has heated up. So you've seen this, you start to sweat. So when the sweat leaves your body, there is something called evaporative cooling that happens in our body. So what happens is when your sweat leaves the body, it will break some of the hydrogen bonds in the water that's found in your blood and thereby cooling you. So again, water plays a huge role in your body temperature. And here's many cool things, the properties of cohesion, which is referring to the attraction of water to each other. The hydrogen bonds are holding the water together. It's called cohesion. Adhesion is the attraction between water and another material. So we have seen this water skater on top of the water because it looks like, see, the hydrogen bonds will make like a invisible pair of film on top of the water. And this leads to high surface tension of water. So water is always trying to occupy a minimal space because of the hydrogen bonding in water. That's why when you spill water, right, it'll kind of bead up on a smooth surface. And because of this high surface tension, insects are able to walk on water and hear something floating on water. And because of the attraction between the water molecules, we see the phenomena of cohesion and adhesion. And this is how water will travel up a tree against gravity. So as water evaporates from the leaf, right, it forms like a chain gang down the tubes of the tree. It's called xylem and phloem. And here, the last picture, you can see capillary action, because water also has a tendency to stick to surfaces. You can see, like, this is what happens when you put a straw in your drink. The water level or the juice level in the straw is higher than the rest of the container. So that is called capillary action. So adhesion, cohesion, capillary action are all vital for water to move up a tree. This is very important, again, relevant to water, which is called as the pH scale, or the potential of hydrogen. It's a logarithmic scale. So if you take water, right, H2O, it can split into hydrogen and hydroxyl. If the amount of hydrogen is equal to the amount of hydroxyl, you call that a neutral solution. For example, the water that we drink from bottles should be neutral. If the hydrogen ion concentration is high, it is called as an acidic solution. Examples would be, vinegar, your salad dressing, orange juice, coffee, all of them are very, very acidic. Whereas if the hydroxyl concentration is high, they call it alkaline or basic. So most of the cleaning solutions are basic. So this is the pH scale. So we saw the importance of water in life. There's no life without water. The next element that is super important in life is carbon. It's very, very unique. So see here, you can see the nucleus and the protons and the neutrons in the nucleus. First shell is filled, 2 electrons. It's stable. Second shell should hold 8 electrons, but it has only 4. So the valency of carbon is 4. And it's this is why carbon is very, very reactive. So the study of carbon is called organic chemistry. All life on Earth is based on carbon. Carbon makes up most of the mass of living organisms. So it forms the skeleton of all the macromolecules that we will be studying. All right, a little bit more about carbon. So here's carbon. It can form 4 bonds, covalent bonds. So when it has all hydrogen, it's called methane. And here's another picture of the different shells. There are 2 shells. It can bond with 4 different atoms. Another thing is it can either make single bonds like this, share 1 pair of electrons, or double bonds sometimes. So it's just versatile. Even triple bonds are available too. And another uniqueness of carbon is that it can form long chains like CH2, 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 like thousands of times. And that is gasoline. So anytime you see carbon and hydrogen together bonded like this, it is a high energy compound. It can form linear molecules, or it can form cyclic molecules like this. So it's very, very versatile. And it can also have something called functional groups. And depending on what it's going to attach to it, the function can change. So now carbon is the basis of all the macromolecules that's found in the human body and like our food too. So we have carbohydrates, lipids, proteins, and nucleic acids. Kindly memorize this little table here. So when you say carbohydrates, it's the energy source for human beings. In plants, the carbohydrate is called cellulose. It gives structure to the plant. So all these are large biological molecules with a carbon skeleton. So they are called polymers. Poly means more than one. Mer is molecule. So a polymer is a long molecule made up of monomers, that is 1 unit. So here's an example of carbohydrate, and here's a monosaccharide, 1 sugar, like glucose, fructose, they are all monosaccharides. If it's 2 sugar, it is called disaccharide, like sucrose is 1 molecule of glucose and fructose. Polysaccharide is a long chain of monosaccharides. So carbohydrates' main function is energy, and in animals and in plants, it gives structure. Lipid is not a polymer. It is made up of two different molecules. Again, it's a very high energy molecule. It also is the precursor for many of the hormones and steroids in your body. It is made up of glycerol, and this long chain is called fatty acid chain. And here's some example of fats, which is triglycerides, and some hormones, which is estrogen, testosterone. They're all lipid-based hormones. Proteins have a huge range of function in the human body. All the enzymes in your body are protein. They can be structural. For example, your hair is keratin protein. Your skin has collagen. It can also have storage function for muscle contraction, right? Your actin and myosin proteins. So proteins have numerous function in the human body. So it will have a central carbon, and then it will have all these different attachments. So we're going to go over all of this in detail in the next few slides. A good example of a protein is the lactase enzyme. You will know it's an enzyme by the ASC, last three suffixes. So lactase will act on lactose from milk to break it down. Hemoglobin, that is found in the red blood cells of your blood, is a transport protein. It will carry oxygen to the different parts of the body. The fourth macromolecule, so protein is a polymer made up of amino acids. This is what the amino acid will look like. Nucleic acid is a polymer made up of nucleotides. The main nucleic acid in our body is deoxyribonucleic acids, DNA, and ribonucleic acid, RNA. So these are the information molecules in our body. So we'll see each one of them in a little bit more detail. So we have carbohydrates first. Sorry. So carbohydrates are the main source of energy in the body, fuel in our body. That's why, like, if we don't eat, you feel tired because you need energy in the form of ATP energy that comes from breaking down sugars in our body for every reaction in our body, for most of the reaction in our body. So monosaccharide is a very simple sugar, and it can't be broken down into smaller sugar. But like disaccharide, you can break it down. You add water to a disaccharide, and then you can break it down into monosaccharides. So this example here is an example of a condensation reaction or dehydration reaction, where two simple sugars come together with a loss of a water molecule to form a covalent bond. It's called glycosidic bond. So glucose and galactose will form lactose. And here you can see when it's shown as a flat molecule, it is called as linear structure. Or you can also see the cyclic representation of simple sugars. If many sugars are attached together, right, like this, if without branching, it is called starch. So this is what you see in pasta and breads. It's very high energy food. That's why you have to mind the amount of starch. The storage form of starch in the muscles is called glycogen. So glycogen will have branches, side branches like this. And another example would be cellulose that is seen in the cell walls, chitin in the exoskeleton of insects. So there's several examples of polysaccharide. Saccharide means sugar. So we saw monosaccharide, that is simple sugar, disaccharide, which is two sugar, and polysaccharide is a long chain of sugar. The next one is lipids. Lipids are hydrophobic molecules, and it will include fats, phospholipids, and steroids. So if you take a lipid, right, it is made up of two molecules. Just memorize all this. It has a glycerol part, which is orange, and then it has a very long chain of fatty acids, CH2, CH2, CH2. So it will have, if it has three of them, it is called triglyceride. So lipids are not polymers. It's made up of two molecules. So the Fatty acid tail will attach to the glycerol part by a dehydration reaction, forming 3 covalent bonds. When you say something is saturated, that means it's saturated with hydrogen. So the triglyceride part doesn't have any double bonds. It is saturated with hydrogen. For example, your butter is saturated. So what happens is when something is saturated, it's solid at room temperature. And these are the ones that are harmful for health because it can clog up our arteries. When something is unsaturated, the triglyceride has double bond. So anytime there's a double bond, there is a bend in the structure of the molecule. See, there's a kink there. And it's because of this that the molecules can't stack up on top of each other. It is liquid at room temperature, and it's better for your health. For example, olive oil is unsaturated. Amino acids. So, proteins. Protein is a polymer made up of 20 different amino acids. So this is the a basic structure of an amino acid. It will have a central carbon. It will have a amino group, NH2 group. It will have a carboxyl group, COOH group, and a hydrogen bond. And then this R is the part where it can vary. So this part is acidic. This NH2 part is basic. And there are 20 different types of R groups. And that's why we have 20 different types of amino acids. And the amino acids will link together by a covalent bond with a loss of water. And it's called as a peptide bond. And there are 4 levels of protein structure. If it is the sequence of amino acids, if you're referring to just the sequence of amino acids like alanine, guanine, cytosine, or whatever sequence it is called the primary structure of proteins. Secondary protein structure is referring to the hydrogen bonds of the peptide backbone. So this will cause the protein to either have a sheet-like appearance, it's called beta sheet, or it will have a helical appearance, it's called alpha helix. The tertiary structure is the 3-dimensional folding pattern of the protein because of side group interactions. So the side group from one amino acid might interact with the side group of another amino acid, and the protein will start to fold on top of each other. Now, why is all of this important? Because structure will dictate the function. And once you lose the structure of the protein, its function is also lost. So that's why primary structure, secondary structure, tertiary structure are very, very important. And when there's more than one polypeptide chain, it is called quaternary structure of the protein. For example, right, if you've heard of sickle cell anemia, one amino acid will change because of a mutation in the DNA sequence. And because of that change, the hemoglobin molecule found in RBCs, right, will change. And because of that, the RBC, red blood cells found in your blood, can collapse on itself, forming sickle cells. So that is why it is very, very important, the structure of the protein. So remember that the bond is called peptide bond here. And the long chain of amino acid is called polypeptide. Okay, the last macromolecule is nucleic acids. So this is referring to DNA and RNA. DNA is deoxyribonucleic acid that is found in the nucleus of the cell, and RNA is ribonucleic acid. So one amazing thing about DNA, we all know it is the information molecule in our body. So it is double-stranded and it is protected in the nucleus. And when it wants to express itself, it will make an intermediate copy called a template, called messenger RNA. And RNA will act as the template for making protein synthesis. And it's the proteins that in our body that is responsible for all the traits that you see in your body. Your hair color is a trait, and that's because of a protein. Your eye color is a protein. So all the traits are responsible, come about because of a protein. So this is how information flows in biology, and it's called as the central dogma of biology. So kindly know nucleic acids are macromolecules. The monomer that makes this is called nucleotide. So nucleotide will have a phosphate group, deoxyribose sugar. So here's the sugar part in blue, and it will have a basic part. It's called the base. So the base can either be adenine, guanine, cytosine, or thymine. We have another lecture on each one of them. So this is only like a overview of what you'll be studying in the future. So nucleic acid, the monomer is called nucleotides, and they make both DNA and RNA. One of the differences sugar in DNA is deoxyribose sugar, whereas in RNA is ribose sugar. DNA is double-stranded, RNA is single-stranded. And the 4 bases are adenine, guanine, thymine, and cytosine. Adenine will base pair with thymine. A will go with T. So these single ring structures are called pyrimidines, and the double ring structures are called purines. So A will go with T, G will base pair with cytosine. So there's 2 double bonds here, hydrogen bonds, and 3 bonds here. So here's the final slide that shows the double helix structure of DNA macromolecule. So the sugar and the phosphate will make the sides of the ladder. The rungs of the ladder are made by bases. Adenine will always base pair with thymine. Guanine will always base pair with cytosine. So this is the base pairing rule in DNA. And I hope that was helpful.